Sir, sorry, very quickly before we start, I have a question about the Morgan experiment from last lecture that I couldn't get a grasp on.
Yeah, please go ahead.
So, if I'm not mistaken, he concluded that if the genes were very distant, then genetic recombination can happen more than once. There's like a chance of interference. I don't completely understand the causality there.
Okay. So let's consider the chromosomes, the chromosomes which are replicated and contain the sister chromatids. Okay, these are centromeres. Now, let's say we have a point A and point B, two different genes, and then their alleles on the homolog chromosomes. Okay, so what I told you, greater the distance, higher the frequency of recombination. So it can recombine at this point, it can recombine at this point, it can get all of them, the result will be A, B. Is it clear? Because
Yes.
this one recombines with this one, and the reciprocal will be this one, AB. Is it clear? Now,
Yeah.
what we learned that the frequency of recombination or percentage of recombination is proportional to the physical distance between the two loci, two different genes. If I bring these two genes, same two loci on the same chromosomes, let's say now A and B become like this. Now, the chances of recombination are minimized. Imagine here you have, recombination percentage turned out to be 25%. Here, it may be hardly 10% because distance is reduced. What actually happens? The chromosomes, when they get duplicated after DNA replication in meiosis, the homologs come together, and these homologs exchange physically the fragments. Imagine B was here and A was there, and A is here and B is there. Now, if they physically exchange their segments, this represents this one will be AB, A, B, and this one will go like this, and they get separated. They get separated, and after separation, you will have AB, and the homolog will be small a, capital B. This depends on, and this structure is called, we call this crossing over, and this structure is called chiasmata. It happens in meiosis. Very, very little chances in mitosis. 1,000 times less in mitosis. So the distance between the two loci determines the frequency of recombination. And when we say 25% recombinants, these are recombinants. And the parentals will be A, B, and A, not A, B there, A, B here, A, B. So parentals will be small b and A capital, B capital. Remember, after meiosis, the biggest numbers you get, they are parentals. In a population, in a progeny, the smaller numbers are recombinants because they are such events are less. When we say 25%, it means in 100 meiotic events, if there were 100 meiosis, 100 cells went through meiosis, 25% will show this recombination because the two distances are 25 centimorgan apart. This is just a physical distance they gave it after Morgan's name. Did I answer your question? Now is it clear?
I think part of it was answered, but I wanted to know about subsequent events, like after the initial genetic recombination. I think he concluded that it could happen again, double recombination events.
Yeah, double recombination can also happen, but that's good that you are asking this. Normally at 101 level, I try to just explain you the simple recombination. So double recombination can also happen. For example, A, B, and A and B. Okay. And double recombination means it recombined here, but it also went back. In that case, it will still be an AB. But double recombination, double crossover, what we call, that is very rare. That is less than 1%. And this double crossover will not be considered as a recombinant. You will not consider it recombinant because you are looking at the phenotype of A and B. You won't be able to document this. However, if you have 3 loci, let's go to more complicated state, which is not part of your syllabus, but just to show what he is saying is right. Let's say we have 3 loci, A, B, and C. Now double crossover happened. It went like this and came back like this. And the reciprocal will be, so what will be the genotypes? Small a, capital B, small c, small a, capital B, small c, and capital A, small b, capital C. So double crossing over also can happen, but everything relies on the distance between the loci. Is it clear?
Yes, sir.
Yeah, there were other hands as well. Who wants to ask question? Amna?
Sir, you know how you said it's possible in mitosis as well, but in mitosis we don't have tetrad formation, right? So how does the chiasmata occur between single chromatids?
No, in mitosis, both mitosis and meiosis, when a chromosome undergoes DNA replication, in mitosis, these chromosomes, we call them mitotic chromosomes or metaphase chromosomes when they look like this. And the homologue will also look like this. But in mitosis, since chiasmata is not formed, so these events are rare. They are very few recombinants, almost negligible. Did I answer your question?
Yes, thank you, sir.
Okay. So the next question and the next big discovery was sex-linked inheritance. Sex-linked inheritance. So by this time, the factors were being called genes. Everybody knew genes, they knew chromosomes, and we started learning it as the whole work from Morgan's lab described the genetic inheritance through a theory which we all know as chromosomal theory of inheritance, in which we learned that genes, they reside on chromosomes, and when genes reside on the same chromosome, we get slight deviation from Mendel's law of independent assortment. genes on the same chromosome can undergo recombination, and we get new combinations, new phenotypes which were not present in parentals, which deviated from law of independent assortment. However, the proof was still not there. And proof for what? What is the question we are? So proof that genes reside on chromosomes, indeed reside on chromosomes. All what we have so far covered, I mean, even in today's examples, we are saying, we have point gene A and B, or A and B like this. It does give us a proof, but something which shows that I have a gene A and this is indeed due to a specific chromosome and nothing else. Imagine nearly 100 years ago, 1920 or so, or later on, from 1920 to 30s, time when Morgan's lab was very active. Now, the answer to this question came, I would say, simply completely finding needle in the haystack, and they found answer, and they found a mutant which was called white mutant. Now, if you have watched lectures where I showed you the flies, normal fly, its eyes are brick red, dark red color. And if you look under microscope, the structure of eye, it's one of the amazing structures in living organisms. Each little, this structure here is an independent eye, and this is a compound eye. But anyway, it's a red color. The phenotype, eye phenotype, is red. Okay. They found a mutant in which eyes were white. And this mutant was named white1118, that allele or allele name or whatever. When they had this mutant, they crossed it with, so let's say we had male, the father white, and female. Normally in genetics, the tradition is that we write mother always on the left and father on the right. So we had crossed white mutant with normal female, and they discovered that all F1 was normal, which means red. So what is white mutant then? The white phenotype, if you get
recessive.
Yeah. So white is recessive to the wild type eye color. Now, what they did, they took F1, this one. They took a female, F1 female. What will be the phenotype of this female?
Red.
Red. And they crossed it with a male which was normal. Normal means whenever in genetics we use word normal, here it is wrong. Normal, it's not, it's F1 red. Whenever we use normal, it means we are referring to wild type, which carries no mutation. Okay, so according to Mendelian genetics, how much they should get? How many red and how many white? If white is recessive,
Reversal.
how much?
3 to 1.
Yeah, according to Mendel, it should be 3 to 1. Yeah,
Won't it be 50-50?
50-50. Someone else also said 50-50 because this is recessive. Sorry, I made something wrong. So this is your white and this is your normal. And how much should be red to white? Ratio?
Sir, I'm confused. You said initially कि normal is red, right? जब हम white mutant के साथ से cross कर रहे थे.
Yeah.
And now normal is white.
Sorry, I did not hear.
So like you said, start में कि white mutant के साथ जब हम cross कर रहे थे, उस वक्त the normal one was homozygous red, right? Like the dominant one, right?
Yeah.
Now you're saying कि जो like जो normal के साथ you've written white. So
No,
like what
I did not.
exactly is the
I did not write white. I say the homozygous recessive based on my F1 result. I give it this number, this symbol, small w. And the normal, which will be red, I give it capital W. You can choose this one. There's no bar that you have to choose W or R. But in flies, there is a convention that the name of the gene like white gene, because this gene was named as white gene. Name of the gene is given after the mutant phenotype, which means the white gene, when it will be mutant, it will give this phenotype, white phenotype. This way you don't have to remember which phenotype will be in a gene X, because the name of the gene X will be after the mutant phenotype. So that's why in flies we give W. And when capital W, it means it's red. It will give you red fly, red eyes. Because wild type तो आपको पता है ना, आप देखते हो रूटीन में, आपको पता है कि उसका क्या. Is it clear?
So like just for clarification, so जो हमारा white mutant है,
Yeah.
that's the recessive one, right? So it's the homozygous recessive, और हमारा दूसरा जो parent है, that's homozygous dominant, which is red, right? Or also, as we're saying here, it's normal, right? And if we cross it, तो सारे heterozygous normal निकलेंगे, like
This
heterozygous
one,
red. Yeah,
F1
this is,
normal, not
yeah, and
normal,
if
F1
we
red. Yeah.
and if we cross this आगे, then we're crossing these heterozygous female red with a male heterozygous red.
No, male normal. Normal means wild type. WT means wild type. Is it clear?
A little, I'm still very confused.
Zainab, is it clear?
Can you hear me? Okay, sir, I'm still confused. I don't get कि like after F1 we're supposed to cross it with the same ones, right? Like the hetero red, but we're crossing it with something else. I
Chale.
don't get.
Yeah.
Right,
It's okay. It's okay. Good that you bring it up. Let's cross it with males with this phenotype. How much red to white you expect?
3:1 ही होगा.
This is what you get. Expect, not get.
Yeah.
And if this is true, if we cross F1 heterozygous with this one, how much you expect?
Then there should probably be no white. Then
No
there
white.
should.
All reds. Now, stay with me here and let's see the results of this on next slide. So F1 red female cross to male normal. What we just said, we should get all reds. Okay. What they discovered that all daughters, they were red, half sons white, half sons red. Clear?
Yeah, but that's because जो मेल वाली जो बेसिकली जो वाई क्रोमोसोम है उसके ऊपर आई कलर की जीन नहीं है, राइट? दैट्स व्हाई इट्स हैपनिंग कि इट्स
नहीं,
ओनली हैपनिंग इन द
अभी
सन.
आप उधर ना चले जाएं। आप अभी पहले स्टेप वाइज चले ना, ताकि बाकी लोग भी सीखें जिन्हें जिन्हें अभी नहीं पता वाई क्रोमोसोम क्या होता है। We said according to monohybrid Mendelian cross, monohybrid, where there is complete dominance. Complete dominance means when two different alleles come together, capital W, small w, one of the two will be completely dominant, and this should be red. You said if we cross this F1 female with F1 male, we should get 3:1. Or if we set up F1 female with normal male, we should get all reds. But what they got when they crossed F1 red female with normal males, they got all daughters red, half sons white, and half sons red. They set up another cross in which they said, okay, let's change parents. And if we use female, previously we used normal male. We say we used all, normal female wild type with males which were white. According to their previous experiment, the very first experiment, they should be getting everything normal because W is dominant. But what they received was all normal. Okay. Did I do something different, or I think I used a wrong parent here. White male, normal female. And oh yeah, there's nothing different here. We use white female, they used white female and normal males, which means red, wild type. What they discovered, all females, they were normal, but all males were white, just right in the F1 generation. So these strange results, where on the one hand they are learning that white is a recessive trait, white is recessive and red is dominant, but the results do not fit with conventional monohybrid cross. And that led them to think that, this is because when we change parents, like here, we, instead of using normal female, which we used in previous example, if we bring now white mutation from mother and father is normal, the inheritance of phenotype, the pattern changes. And they said the inheritance of white shows sex-linked inheritance, because but it matters if the phenotype is coming from mother or father. If it was from father, all F1 was red, and this was a puzzle for them. And whenever scientists, they don't have an answer, they try to read they normally read a lot. It's expected that, you know, we read at least, we are up to date with literature, whichever latest paper is coming in our field. But whenever we come across such a big puzzle like they came, they went through literature. If someone has described something sex specific chromosomes, and they discovered that Nettie and Stevens, I think it was 1905, they, when they were looking under microscope, they described that whenever they looked at gametes, female and male, they could find that, you know, the female gametes, they had same chromosomes, while the male gametes, they show heterogametic situation in which, you know, one of the gamete is always showing a chromosome which is different. So let's say if it is fly, and normally you have you know 4 chromosomes. So one of the chromosomes is in males, in gametes, let's say a diploid underwent meiosis. So when the 4 gametes were there, the 2 of the gametes were, I'm just not having other chromosomes. Let's say like this, we had chromosomes. So they named these chromosomes, and in females, all the chromosomes were of the same shape. What they discovered that in somatic cells, in diploid cells, males versus females, in the females, all the chromosomes were having perfect homologs. Okay, they all were, let's say, like shapes like this, all 4. But in males, we had one of the pair was always like one of the chromosome was of a different shape. They named them X and Y chromosome. In females, the name said, okay, the one which is having homolog, they are XX. Now Morgan's lab said, our Y gene, it could be on the X chromosome. Now, let's say we used normal females crossed with white male. Now let's assume we had females, normal is white, and male is going to show you like this. This is X and this is Y, this is X and this is X. In F1, now in gametes, the gametes will be, this will be 1. All 4 from a diploid will be like this. But this one will be small, not capital W, sorry, small w, white. And Y and Y. Now, any of the gamete, if we draw the like this, WW and sorry, XW and XW, and here this one XW and Y. Now these are the female normal allele, capital W. So this will be capital W and then small w. Capital W, Y. Capital W, small w, capital W, Y. So that's why in F1, we get all normal. But when we crossed F1 female, which was red, these ones, with a normal male, what we obtained was all daughters normal and half sons normal, half sons white. Now use this. F1 red is what? Female, both X, but it's heterozygous. And normal male is what? Normal male is capital W, Y. Males carry Y chromosome, we said. Now, if we again draw this thing, capital W and small w, the mother gametes, and the father will be normal and Y. Now this one will give you capital W, capital W, capital W, Y, small w, and capital W, and small w and Y. Now what will be the sex of these four? Gender of these four? What do you predict of this one?
Female. Female.
Female. Why female?
Because 2X chromosomes.
2X chromosomes. This one?
Male.
And the phenotype?
Red.
Red. This one, female red. What will be this one? Male or female?
Female red.
Female red. And this one?
Male white.
Male white. So isn't it this one? Half sons normal, half sons white, and all daughters normal.
Yes.
So this led to the discovery that if I use, let's say, a different one now, if I use female X chromosomes, because we know female is with 2 X chromosomes, and we also learned that white is a recessive trait, which means it has to be homozygous. And if we have single X in male and then Y chromosome, the F1 will be, all daughters will be showing such phenotype, but all sons will show you phenotype either this X or this X will gamete with this X or gamete with this X. When it gets fertilized with this one, all males will be what will be their phenotype?
White.
White.
Yes, this is white and all these are red because it has to be homozygous recessive, small w to be white. So this is dominant allele. If you now cross F1 female with F1 male, what you get? You get gametes will be this capital W or small w, 2 gametes of this, 2 gametes of this. And here you will get small wx gamete, 2 gametes of this, and 2 gametes with Y. Now, if we write down all the possibilities, so you will get capital W with small w. You will get in the progeny small w with small w, this one, or previously this one we wrote like due to fertilization of this, or we'll get small w with Y or capital W with Y? So Shamin, you tell me what is the phenotype of each of this individual.
So the first one is red.
Good,
The second one is white.
good,
Third one is white.
good.
And the fourth is red.
Excellent. So it's very important to understand when we have small wy, because there's no dominant allele, the homolog is missing, this will demonstrate or exhibit its phenotype, which will be white. That's why sex-linked mutations, if they are lethal, they can be lethal in males because we lack one of the homolog. And our probability to die with, if it's a lethal mutation, is much higher. If we inherit that, if we are carrying that mutation and that's lethal, we will die. In human females, it's still, if they carry, let's say, mutation A, and this is the mutation, you know, they still have one normal copy and they may survive. Okay. Now, these chromosomes, X and Y, they were called sex chromosomes because genes, I mean, we just looked at the eye color phenotype, but what we have learned in next 100 years till today is that there are genes on X and Y which play a role in sex determination, whether we will be a male baby or a boy or a girl. In case of humans, for example, in humans, there is, let's say I write down,
I have a question.
Y chromosome. There is a gene called SRY, which is on the Y chromosome, and that is responsible for maleness.
So I have a question.
G.
Just want to tell that since the X chromosome is larger than the Y chromosome, if someone has a daughter, does it mean that if someone has a son, sorry, does it mean that they share more genes with their mother? Like just because they're inheriting the X chromosome from the mother.
Please speak aloud a bit. I could not understand.
I wanted to ask that if someone has a child that's a boy, does that mean they have that they share more genes with their mother?
No.
Because the X gene is larger and has more genetic information.
No. So look, boys will have X and Y, and girls will have X and X. 2 X chromosomes. So the girls, this is father's copy X and this is mother's copy X. And boy has, in this case, the gamete from father, which was fertilized, is carrying Y chromosome, and this is mother's X chromosome. Okay, clear? So there's no that boys carry more or girls carry more. You know, this is a more specialized topic. We call this X chromosome inactivation. It's a very interesting study. I mean, go and read about this. This is something interesting. So why it is interesting? Because the boy cells, the male cells, all the cells contain just one X chromosome. And female cells, all the cells in their body contain 2 X chromosomes. Now it means human cells will be having here, let's say, the gene dosage or amount of proteins of all the genes which are on the X, they are two times more than male. So in human females, very intelligently, they inactivate all the genes. The whole chromosome is shut down. One is literally jumbled up. Everything is silent, inactive. Look how much they care for us, because we carry just one X. If both the X chromosomes are genes on the X are active, this is an abnormal life and death is there. Okay. Y contains very little genes, very, very little genes. But most important gene is the SRY, which is responsible for maleness in humans. And all the Y chromosome, it's not at all homolog of X. It's in terms of its structure, the genes, everything is different than the X, except a very tiny part of Y. Clear?
Sir, I have a question.
Ji?
Sir, you said the Y chromosomes of the male contains SRY gene. So, sir, do जो female का जो X होता है, do they contain any type of gene like SRY gene या उसका कोई alternate या they don't contain anything like that?
No, they don't have SRY. But if we
Any
go to fruit flies, there we do have sex-specific genes. And
okay.
okay. But let's.
Sir, I just wanted to add something.
Yeah.
So the thing that you said about one X chromosome being inactive, this creates a pattern of stripes on every female, right? And this is why all calico cats, which have different colors of like different stripes, they're all female because of this reason also.
Excellent. So the calico cats, because this is random. So, and this happens very early in development when females are just, few cells. I exactly don't remember the day after fertilization. So let's say fathers copy X, mothers copy X, fathers, mothers, father, mother, father, mother, father, mother. This is what the genotype of human females is. And remember, this is besides the other 22 pairs of chromosomes, which we call autosomes. X and Y are called sex chromosomes. Other 22 pairs are called autosomes. And here we are just writing X and Y, or in case of females, both X. So this is a random event whether father's copy or mother's copy will be active. In some cells, it could be father's copy is silent. In others, mother's copy is silent. But once it is silent, the mitosis of this cell, let's say father's copy was silent and mother's was on. So the father's copy will be inherited in the on state and mother's copy will be on. When this will go mitosis, through mitosis, mother's copy will remain on and father's copy will be off. Okay? Mothers will be on, father will be off. Mitotically, they go like this. Now, she's asking calico cats, and if you know the genes, that is basically those patches which you see on the calico cats, So there are, these patches. That is due to, which of the cells are contributing to which region of the skin in the body. And if the mutant, the cells which contain that gene is on the chromosome which is switched off, then you get these patches. Yeah, it was very good that you highlighted this. I wanted to just one last thing wanted to highlight. In both humans and fruit flies, this is the genotype of males and females. Homo sapiens, humans, and Drosophila melanogaster. However, if you get XXY, in humans, it's a male, but in case of Drosophila, it's a female. And if you get XXY, and what is the other?
X0 or XO.
X0, which means lacking Y. Okay. So in this case, in this will be a female, and here it will be a male. Why? Why this discrepancy? Because of presence of Y, there will be SRY. In humans, it's a male. In case of females, in case of Drosophila, they say the ratio between X and Y. So Drosophila has 4 chromosomes. So we write them like this: XY, autosome 2, autosome 3. Normally we don't write 4th chromosome. So we just consider there are 3 autosomes, chromosome 2, chromosome 3, and 4th, but 4th is so rudimentary and heterochromatic, we don't write it. So we say so how many X? 1 over 2 autosomes, ratio is 0.5. This will be a male. And if you have XX and 2 autosomes, so 2X over 2 autosomes, ratio is 1, we say it will be a female. Actually, the underneath phenomenon is, I told you, there are genes on the X chromosome and these genes are called SXL, sex lethal. These are the name of the genes. Then in males, there is a gene MSL. These are the name of the mutation, male sex lethal. So these genes actually reside on the X chromosomes, and they are involved in sex determination in fruit flies. That's why you see a slight discrepancy when you have X0, single X. That's why it develop as a female. Absence of Y actually results in female here in the humans. So in the recorded lecture, there is also one last thing, which is the incomplete dominance. We haven't gone through that, and I want you to just stick to simply the complete dominance examples we have covered so far. Incomplete dominance also was later on discovered that some of the genes are not completely dominant. For example, you cross red with white and you get pinks. And when you self-cross pink colored flowers, plant, you get, reds, pinks, and whites. And they said, okay, let's assume this is red, this is white. So when the 2 different alleles are present, neither this one completely dominates nor this one, and we get an intermediate. And when this will be self-crossed, there will be both homozygous will be red, heterozygous will be pink, and homozygous will be white. This is also present in plants. So there is incomplete dominance also present, but just stay on what we have covered so far. And what also you should try to do is you should try to go and solve problems where you can make a genetic map or measure the distance between 2 genes on chromosome based on percent recombination. If you go, there is an example at the end of the recorded lecture where you have, I told you, the biggest numbers you always get parentals. And the small numbers, let's say 450, 460 are parentals. And then the recombinants you get like 25 and 30 or so. So you will simply take these recombinants, 25 plus 30 divided by total, which is all parentals plus recombinants divided by multiplied by 100, you get percent recombination, and you can learn about the genetic maps. So we have the quiz today. Abdullah, are you there? Is your lab done?
Yes, sir. No, sir, I just came for the quiz.
Okay, please switch on the activate the quiz.
So I have just published the quiz. Go to LMS जहाँ पे वो हमेशा होता है, and you will get, it's only one question and you will have 1 minute to solve it.
And your exam is going to be on 24th. I sent you an email. So your exam is on 24th at 7 in the evening, because 23rd is an official holiday. So I sent you an email already. 24th March, Wednesday. Allah Hafiz, good luck with your quiz.